Hello, pharma professionals, students, academicians. Welcome to Pharma Tutor Expert Live on YouTube. See, our country is slowly and gradually coming out of pandemic of COVID-19. But one positive change came out of this pandemic. That is, it is easier to get connected with national and international talents or speakers, and we are witnessing globalization in the exchange of knowledge, which become very easier and it become with better ways. Well, our today's event topic is pharmacogenomics and personalized medicine, which is jointly organized by PharmaTutor and International Pharmaceutical Forum (IPF), which is glittered by the presence of Vice President of PCI, none other than Professor Dr. Sailendra Saraf, who needs no introduction, and along with them, we are accompanied by national international speakers from various parts of the world. And believe me that today's topic will not only enlighten you with the knowledge, but it will also change your thought processes about how modern era of medicine is changing so quickly. So I will just introduce you guys with pharmacogenomics terms. So maybe few of the students are also there in this session. So before going in details, see, pharmacogenomics is simply the study of how genes affect person's response to drugs. It combines pharmacology and genomics. Although it is common to see pharmacogenomic testings in US and Australia or Canada, but it is not yet part of practice in India. So do you know that there are already established pharmacogenes are there, just like cytochrome P450, then drug transporters are there, human leukocytes are there. So these are already established pharmacogenes are there. And I'm not going in deep in that, but there are more than 15 medicines available with proper pharmacogenomic prescribing in USA. These medications are also available in India, very freely available. Just like I'm giving you some example of atorvastatin, metoprolol, warfarin, codeine, etc. So these medicines are already there with pharmacogenomic prescribing guidelines, and there is a serious role of pharmacists in establishment of pharmacogenomic testing model in the countries like UK, Canada, and Australia. Well, let's move up with our session. Before inviting our chief guest, Dr. Saraf sir, Dr. Fayasudin from IPF will address our welcome note. Welcome, sir.
Good afternoon, everybody. Thank you, Dr. Rajesh, for giving me the opportunity to welcome our chief guest speakers. First of all, it's a great honor for me to open the Pharmacogenomics and Precision Medicine International webinar this afternoon. I think we are more than 1,300 participants here live streaming on this channel. It's a great honor for us within a short period of time. I congratulate Dr. Rajesh. He has gathered such a nice opportunity for all of us with a beautiful title of Pharmacogenomics and Precision Medicine. I am happy to greet all of you here on the behalf of organizer and host of this program, and also extend a warm welcome to all esteemed attendees who have registered here from diverse places across the globe. We are delighted to have everyone join us here today. I hope that you will take the time to learn and interact with our esteemed speakers. Once again, thank you very much for your time. A very, very special welcome to our special thank to our chief guest, Dr. Sailendra Saraf. As Dr. Rajesh told us, there is no need of explanation how much the personality he is. He is the Honorable Vice President of Pharmacy Council of India. I am very much thankful to you, sir, for accepting our invitation and joining us today here at this platform. Your presence will make a lot of difference to the young minds attending this satellite session. Despite of having a very tough schedule, you have given us the time. We are fortunate enough for your presence here, and I'm sure that this program will be going to be very recognized, and people will enjoy this session. Coming to the personalized medicine or precision medicine, it holds the promise as Dr. Rajesh told, that treatment will one day be tailored to your genetic makeup. It's going to be the future. We are the pharmacists, and it's our leading role. When we talk about the internet, there was a 2G, 3G, now 5G, and most of the countries are opting toward the artificial intelligence and machine learning things. But why not in medicine? We are just taking the kara, we are just taking the traditional things, we are opting somehow for the novel drug delivery system. But when we talk about the genes, the genome, we should keep in the mind that the pharmacist role is a very, very enthusiastic, it's a great potential, great role here. Actually, the modern medicine or modern medication save millions of lives a year. Yet, any one medication might not work for you, even it work for other people. There is a severity of things. There are many, things are there. Your age, lifestyle, health, all influence your response to the medication. But we lack the thing that is our gene. Genes are also responsible. The races. So pharmacogenomics is nothing but it's a study of how a person's unique genetic makeup, that is genome, influence his or her response to medication. And the precision medicine aims to customize healthcare with decision and treatment tailored to each individual in every way possible. I'm sure this webinar has been basically designed and developed to introduce you to some of the concept of personalized medicine, which we'll be discussing during the course today afternoon, this session. We have a very good speakers here. They are eminent, they are expert in their field, they are already working in many hospitals and industry. First of all, I would like to welcome our first speaker, is Dr. Mohi Iqbal. Recently, he is the associate professor in pharmacology at Taibah University in Medina, Saudi Arabia. He will give us a very wonderful talk on pharmacogenomics aspect in COVID-19 treatment. Actually, COVID is going on, and he will he will enlighten the possible treatment options on COVID treatment in special regard to the pharmacogenomics. Our next speaker is Dr. Meraj Alam. He's a research scientist at Hospital of Sick Children at University of Toronto, Canada. He will give a talk on role of neutrophil extracellular traps in COVID lung pathogenesis. And our third speaker is Dr. Nitin Saigal. He is a senior scientist and a team leader at Hilleman Laboratories, India. And he will talk on microneedles as a transformative substitute to conventional parenteral delivery. I hope that all of you will have a most productive time of interesting and stimulating session. I sincerely wish this international webinar come expert discussion will be a rich success, not only as an opportunity to share knowledge and expertise in pharma education and research, but also as the beginning of a long, fruitful cooperation and friendship among the fellow educators. Last, I again extend a warm welcome to everyone present here. May Allah subhanahu wa ta'ala bless us and accept our efforts in making this earth most beautiful and safe to live. Thank you very much. Thank you, everybody.
Thank you. Thank you very much, sir. Thank you, sir, for welcoming our chief guest speakers and audience. Now, as I told earlier, that our chief guest, Dr. Saraf sir, is a well-recognized pharmacy professional in Fraternity of India, Pharma Fraternity of India, and he's full of positive energy that if you will interact with him for just 5 minutes, you will ignite with fresh thoughts. Yes, he is a vice president of PCI, and his experience is so deep and rich that he had completed his MPharm in the year when I was actually born. So this is my honor, and it's a really honor moment for me, emotional moment for me, that I am introducing such a personality. He served as professor and principal at BRN College of Pharmacy, Mysore, MP. Later, he joined in Pandit Ravi Shankar Shukla University, Raipur as Dean. He was served as the chairman, Board of Studies, Pharmacy, and VC of Durg University, Chhattisgarh. During his career, he's associated with many statutory and regulatory bodies like DTAB, AICTE, DST, UGC, NASC, NBA, etc. So many he is associated with so many regulatory bodies. Also, he has received so many awards that there is a big list of it. But I will just highlight main out of that: that International Institute of Organizational and Psychological Medicines, USA, Australia, UK in 2019. He also received IASTM from Dr. KR Parikh Award. Also, many awards from API, IPA, IPC. It's a big list. So, with these brief introductions, I just inviting sir to guide us ahead for this international webinar.
If I am visible and I am audible also.
Yes, sir. Yes, sir.
Okay. Okay. So, very good afternoon to all of you. And my dear friend, Dr. Bag, and today's speakers, Mr. Nitin Saigal and Mr. Iqbal, all other members of organizing committee, and most important, the fellow pharmacist online today for today's event. I am really happy, and it's my pleasure. At the same time, it's a definite honor for me to join your platform today, because it is jointly organized by Pharma Tutor as well as the International Pharmaceutical Forum, and it's really important. And you have taken a very befitting topic, and I am really happy for different reasons, and this is very important also, because just after the corona, we were observing several webinars, and we are educating ourselves. But most of the time, the topics of those webinars were very, very generalized. So I'm happy that this time you have selected a very, very specific topic, and that too you have invited the speakers, those who are already into this type of research. At the same time, while I'm talking about this topic, this is somehow addressing the contemporary needs of the academic and research fraternity. But the most important for me is, because as Albert Einstein always said, the research is the highest form of the ideation is the highest form of the research. So such type of activities, this is really going to be the food for the brain, and it will give the scope and platform for the ideation for next years to come. So my compliments to the organization, particularly Dr. Rajesh Bag, for organizing such a beautiful program and selecting this important topic. While we are talking about this post, we can say we are just in the pandemic era, but still I am considering as a post-pandemic era, because we have learned how to remain healthy even if you are affected with the corona. So this is the heads up to the scientists and medical fraternity, that they have developed the protocols and identify also the parameters which are going to affect the health. So this is one of the example itself, that a virus has come, and before vaccine developed, we have developed the protocols. So this is time for all of us to pat our own back, but at the same time, we are bound to think about the overall scenario of healthcare in this country. As far as India is concerned, this country reflects a real paradox in its activity. At one side, this McKinsey has 2 years back identified, and it was they claim that India is the preferred destination for the medical tourism. It's true, we have a lot of good facilities, but at the same time, there is a large number of people, and sector of the population is not having accessibility and affordability of the drugs. So this is the paradox we have to always keep in mind while we are talking about the policy and future course of action. At the same time, while we are talking about the better facilities, so somehow they are centralized to some big places or metropolitan cities and all that. All such activities are not generalized. And particularly this pandemic has given the acid test for the assessment of this healthcare infrastructure and qualified manpower is concerned. So we have understood that we are not at all prepared. We were not at all prepared, and we are not at all prepared to face the pandemic like this COVID in future as well. So there is a need to have a huge investment and a clear-cut roadmap in order to achieve those goals if we really want to cater the needs of the society. As far as the manpower is concerned, so we have demonstrated, at least pharmacist has demonstrated, that without any training, without any advisory, and without any systematic documentation, they have transformed themselves overnight in order to cater the needs of the patients. And this is also one of the important, I salute all the fellow pharmacists, that there is not even a single observation throughout the country that pharmacists were found involved in unethical practices. So this is the one of the greatest opportunity was there for before the pharmacists, because for last 70 years the pharmacists of this nation were struggling to get the recognition. Thanks to COVID, thanks to this pandemic, and thanks to the competent pharmacist frontline warriors that they have brought this laurel to this profession, we are recognized, and now we can claim that we are the real member of the healthcare team in this nation, which is accepted not by the society but the fellow members of the healthcare team as well. So this is something important. At the same time, when I'm talking about this health infrastructure, so still there is a lot of investment. There is a lot of investment while we are importing the medical devices. Even 85%, more than 85% of devices we are importing from US alone. At the same time, we are in importing a lot of intermediates and bulk drugs from China. But again, these two areas were identified by the government of India, and Department of Pharmaceutical has come out with a very lucrative, a very attractive scheme of performance-linked scheme, that is to promote the self-sufficiency in the field of bulk drug intermediate as well as in the field of medical devices. So at the same time, when you are talking about the this manufacturing of drugs, so we have considered that our country has supplied drugs to more than 150 drugs even during the pandemic. There was a cash flow problem, there was a logistic problem, and one important observation we have noticed in those days, when there was a problem in the supply of the raw materials and all that. So the industry of Gujarat alone has developed and rejuvenated their own industry, bulk drug industry, and they have somehow made the this up to some extent self-sufficient for the production of those drug molecules. So these are certain things which we can say we have achieved, but still there is a lot to be done in years to come. And we have to while we are living in a society, typical society, so this is need to understand the dimensions and we can say coordinate of the society. And the most important factor I can we can discuss about the demography of this country. It's really great country, and this is a lot of changes. Socio-economic factors are there, where there is a, as far as investment in health is concerned, so we are far behind as far as the developed nations are concerned. We have very small investment, and at the same time we have very low paying capacity, average paying capacity in the healthcare. So all these things we have to keep in mind. At the same time, we have observed a lot of hardship in the regulatory framework also. This we have suffered just in case of clinical trials, because the world was looking, because of this diversity, to have the phenotypic variation based clinical trials, and just because of the non-availability of appropriate mechanism in the regulatory system, we have lost the opportunity of clinical trial market in this country. And at the same time, when we are talking about this healthcare profession, so we have to again underline two important things. One is the change of we can say disease profile. While we are suffering from infection based diseases, so after this after the 30 years of independence, this profile is shifted from the infection-based disease to lifestyle-based diseases. So this we have to keep in mind. At the same time, we have to again keep eyes, because the pharmaceutical industry is included in the greenfield investment. So there's a lot of FDI is coming out, and if we want to really take the advantage of this pharmaceutical industry, so we have to come forward and we have to make ourselves self-sufficient with the investment in this particular area. While I would talk about, because you have taken a very important topic, that is customized medicine and this pharmacogenomics, but we have to understand the overall scenario of this country. Like, why I am saying so? Because in 1950, a concept was coined that is known as chronobiology, and it was again considered as this will help to develop the personalized medicine, because each and every individual have different biological clock based on the activation and deactivation of pineal gland. So based on this, different methods have been developed, but looking to the shape and size and socio-economic conditions, we could not adopt the development of this science. So whatever the science we are going to develop, or we are going to develop the products, we have to first understand what is the acceptability, what is the affordability, and what is the effect of those. So we have to always carry out the risk assessment and profit assessment. Then only we should come forward as far as the research concerned. As far as the world trend are concerned, so there is a lot of work going on in the field of artificial intelligence. And to everyone's surprise, the Watson, IBM Watson has already more than 40 pipelines are there, and they said within 2 to 3 years they will be there in the market. They will going to command the market in a big way. So we have to keep in my mind. At the same time, I was also discussing about this chemical space for discovery of the new drug molecule, and believe me, India is the really we can say a treasure island, where we can have the good blend of traditional knowledge, Ayurveda, and any system of medicine in the form of different therapies available. So there is a need to have the translation of those therapy. And if we are really coming out with those in development, then we'll be able to have the respect to command in the society. Another area that is commanding nowadays is RNA-based small molecule to uh, treat the diseases. And some of this topic is directly linked with this. And it seems like a smart strategy to start a earlier identification of drug, and through the this gene targeting and gene triggering. These things are possible. But for this particular thing, there's a need to have a use that the creation of huge data with the help of big data. Then only you will be able to segregate the population in different genetic groups. At the same time, there is need to have methodologies. There is need to have certain markers to identify different groups in the genetic scenario. And we have to keep always in mind, we have to keep in mind that is, it should be affordable, it should be fast, it is rapid, and it is reproducible as well. So there's a lot of work is required in this field, and without the help of big data, and without have the artificial intelligence, this must be a difficult task to achieve. Some other points I have noted down, like new drug discovery. More than lakhs deaths are occurring in Western world as well, just because of the antimicrobial resistance. And everybody understand this, because of this uh, the discovery of antibiotics is not very cost effective. It because if you want to develop a antibiotics, you have to carry out the clinical trials on each and every microorganism. Then only you can claim your antibiotic is foolproof. So after 1970, only one antibiotic, full-fledged antibiotic, has been discovered, and it is threatening really for the human breed, because one of the thought leaders has said, somehow with the ever increasing this microbial resistance, we are heading towards pre-antibiotic era. And we all have, it is not a for long, but it's we have really clear in our mind and in our memories what was the impact when there was an infection in the pre-antibiotic era. So if we have to really work in the field of pharmaceutical sciences and we really want to cater the needs of the society, then we have to simultaneously work on two things. That is how to prevent the microbial resistance, at the same time discovery of new antibiotic in order to save the human race. And big database phenotyping screening for the development new drug molecule is also emerging like anything. So again, this is for this type of activities, this type of drug discovery, India seems to be you can say a green field to work in this area. Then there is a concept of fast and rapid pharmacological clinical organ on chip concept. And recently it has been observed that they are developing a chip which is representing the whole body. So they are going to the scientists are going to make the skinning is very fast, but still they are having some limitations. And as Wag has mentioned, there are certain enzyme system like cytochrome for 450 and all that, which cannot be translated in the in vitro in situ activities. So still there is a limitation, but they can make our research analysis very fast. At the same time, a few countries have developed the this bio ink to develop the this bioprinting of some cells, tissues, and organs. And nose and ears are easily nowadays printed through these devices. So 3D printing is also one of the important aspect, we can say. In this area also we can work, but in the field of overall research, there is a need to have ideation based research, because we are patting the back for having the supply of the drug to the world, but real heroes emerged out of this time where the simple three-layered mask and sanitizers. So we have to keep in mind, because we had a very focused this whole of the nation was focusing on the formulation, manufacturing of formulation. All other fields were ignored. And believe me, the electronic this devices and medical devices have commanding the respect in society. So there is a need to have a proper orientation of the research in the field of medical devices and electronic area also. Because if you're talking about the this personalized medicine, so somehow we are again I mean, going back to this compounding and this dispensing of doses form, because we want to give the specific drug in a specific molecule, and there is need to have a steady, proper studies of the genetic structure and in the total information of a a particular person. And this is next to impossible looking to the youth population. So there is a need to have disaggregate those patients in different groups. But when we are talking about this drugs, one more area is also emerging. This is known as a micro samples for the analysis. Again, there are certain limitations. But this is going to have the great impact as far as the discovery of new drug is concerned. But at the same time, when I'm talking about the development all such fields, so there is a need to work in collaboration with other we can say representative from other medical health group. So there is a need to have a translation research with the help of multidisciplinary approach, because isolation is leading to because this specialization and super specialization leading towards the isolation. So when we have to translate the academic research in the commercial value, so we have to adopt the concept of multidisciplinary research. And for this, there is a need to have multidisciplinary communication across the health professionals, which is missing in this country. So this is immediately required. And as far as mine is concerned, what future I want to see in years to come is a bit different from others, because I want to see this country as one of the nation, because world is recognizing India as a pharmacy of the world. And even our Prime Minister also mentioned that this is the asset to the world. At the same time, this is pharmacy of the world. But this is time we have to think about what is next. And for me, at least for me, the next is we should transform our country into a laboratory of the world. And that is possible if we are really focusing on the development discovery of the new drug molecule, indigenous drug molecule. Then we will be able to command the respect, but at the same time we will be able to command the financial gains as well. So this is my understanding. At the same time, capacity building a pharmacist working in different facets of the pharmacy should be an ongoing process, because I want to see in my lifetime pharmacies in the pharmaceutical profession of this world, this country is transforming into a laboratory of the world. Pharmacist is converting a commanding respect as a full-fledged, competent, professional professional. I want to see the drug stores of this country as a full-fledged pharmacies where consultation is also available. At the same time, we are looking forward to have the academic institution which are frontline research institutions and joining head with the industry and giving the befitting solution to the society. So this is only possible with the help of the activities like this brainstorming session on personalized medicine and pharmacogenomics, because every time we have to understand what is going all around. If you are aware what is happening all around, then only you will be able to draw a roadmap so that you will be able to reach where you want to reach. So this is very important, and I am really uh, thankful for inviting me. But at the same time, my compliments to the organizers for taking this befitting topic and the right time organizing this activity. And I am sure this is not the end, but this is simply end of beginning. You'll keep on organizing such type of activity, very specialized, focused, and at the same time, I have personally an expectation that you are going to publish a report and you are sharing with different bodies what are the recommendations based on the deliberations of different experts. So it must be a real contribution from your side for making this profession valuable, respectful in the world. So my compliments to you, Dr. Rajesh Bag, the organization Pharma Tutors, International Pharmaceutical Forum, and I'm thankful to all the fellow pharmacists. You have those who have joined, and I'm really proud of all of you, because I was really I cannot tell you how I was shocked when it was pending was announced, but today I can say I am really how much proud I can I also not explain, because without any problem you have demonstrated that you have transformed from your conventional approach, conventional pharmacist to an international pharmacist, a conventional teacher to a conventional international teacher. And even students of this country, they have transformed from simple classroom teaching to international learners, and that is the purpose. So while we are suffering from this pandemic, some has paid the high value during this pandemic, but there are leaders who have set the new standards, new heights to follow in the society. So once again, I am wishing a grand success of this event, and I am wishing all of you stay healthy, stay happy, at the same time stay active, and stay keep on contributing for the betterment of this pious profession. Thank you very much, and once again, thanks for inviting me and providing this opportunity to interact with my fellow friends. Thank you, Dr. Rajesh. Over to Rajesh.
Thank you very much, sir. Thank you for guiding us to give us a proper scenario. I think we got a overall scenario of the Indian pharmaceutical industry from your talk, and it was really appreciating for and it's an honor for us that you have accepted our invitation and gave us time for this. Thank you very much, sir. Thank you. So we will move ahead with our next session. Now we will go on the technicality of the today's speech, that how exactly pharmacogenomics is working and how research scientists are working from all over the world who have already joined with us. So the first session is from Dr. Mohi Iqbal. Dr. Mohi is pharmacist, pharmacologist who had completed his PhD from University of Sydney in year 2008. He has worked in Westmead Hospital, New South Wales, Australia, as a clinical trial coordinator, and then moved to academia in year 2009. He's currently working as associate professor in Department of Pharmacology and Toxicology at Taibah University, and his theme of the talk is pharmacogenomic aspects in COVID-19 treatment. So here I am connecting you all with Dr. Mohi. Hello, sir. Welcome, sir.
Thank you very much, and welcome all. Thank you very much for organizing this thing. I'm very blessed to be here on this platform. Um, so, in the interest of time, also, the nature of online things, so I'll keep my talk as concise as possible. At the same time, not losing much of the information too. So, I'll start with sharing my screen before we get into. Okay. All right. So hope the screen is on. Do I get some comments here or?
Yes, it is started.
Yeah.
Please, sir. Yeah.
Hello. Yes. Okay. Okay. So, so I'll talk about the pharmacogenomic aspects in COVID-19 treatment. To be honest, when I was looking around for specific for the treatment in COVID-19, I couldn't find much pharmacogenomic aspects into it. So whatever I was able to find it, I'll just present it to you. But to start with, because the drugs which we use in COVID are not, most of them are not narrow safety margin drugs. So the aspects of pharmacogenomics may be not that prominent. But yes, there are so many different cases, so many different millions of patients with different scenarios. So yes, we need to know about these concepts, and these are the real situations that can the clinicians can face in their practice as well. To start with, so what is pharmacogenomics? So here we have two different terms, pharmacogenetics and pharmacogenomics, which are more or less used interchangeably. And they represent the relationship between the variation in gene. So when we talk about one gene, that is pharmacogenetics. But when we talk about group of genes, that's where we mean pharmacogenomics. So that's the variability in genes to variability in drug response. When you talk about PD effects, which drug disposition, when we talk about pharmacokinetic effects, that is variation in pharmacokinetic effects, or it could be related to toxicity as well. And to be honest, I'll say pharmacogenomics is one of the confounding factor in precision and personalized medicine. There are so many different factors that can affect the effects of a drug. It could be age of the patient, it could be gender of the patient, it could be the disease state of the patient. So there are so many different factors. So pharmacogenomics or genetics is one of the factors that can affect the drug effects in a patient. So when we have a genomics thing, so if you want to divide, say, people into based on genes and the effect, so we can divide the people into 4 different categories. So one of them is we gave them the normal dose we give, the therapeutic dose which we normally use as a standard dose. So that's like the full dose or the standard dose. So that is targeted for the majority of the people. We normally say they are the wild type people. It doesn't mean they are wild, but they are the wild type, which refer to the general population. So these are the ones we give them the normal dose. But there could be some patients, they might need half of the dose or maybe a different dose than what is the full dose is. And there could be some patients which are actually need a more dose. So they might need double the dose, sometimes even more than double the dose. And there could be some kind of patients in the group of patients which would not even respond to the treatment, or they might have some adverse effect to that, the same therapeutic dose. So maybe that particular dose or drug is not even suitable for those patients. So based on the gene of a patient or a subject, and in relation to the drug effect, you can divide them into 4 different types. wild type, the normal patients. And let me tell you, majority of the people, about 85 to 90% fall in this wild type category. So when we're talking about precision medicine based on genomics, we're actually targeting the rest variant type. We're targeting the 15% of the patients. Okay. So based on the gene treatment, pharmacogenic based treatment, especially we say gene to drug pair. So which gene is affecting the effects of which drug. So based on that, the FDA recommends like following 4 different things to do with the test. So in some gene to drug pair, FDA would say you have to do a test. So testing is required before even you start the treatment. So that probably may be to pinpoint the patients which might be helpful for that particular treatment, or to avoid certain patients who might be at a risk for some special adverse drug reactions. We'll come up to some of the examples to just emphasize these things. Then the other recommendation which you might get from FDA is like you say, testing is recommended. So in that scenario, is like, it's not like the testing is required, but if you do the testing, you might be in a better position to give the right dose to the patient or choose the right patient for a particular drug, choose the right drug for a particular patient. And sometimes that could be an actionable pharmacogenomic test. So what does it mean is like the interaction between gene and the drug is significant, but it doesn't come to the level where it is like a requirement. It doesn't come to the level. It might get significant or more profound when you actually, in some other situations like drug-drug interactions. So for example, there is 2 different drugs and a gene playing that in that scenario, perhaps it might become even more important. So having that information is always good to take the informed and evidence-based practice. Then the third category could be informative pharmacogenomics. So where this is like for that particular gene and drug pair, so usually the outcome is not significant. Maybe it's not clinically relevant. So maybe you don't even have to perform the test. But of course, if you have the information, you can use it for something else. But when it says the testing is required for any gene and drug pair, that's even becomes some legal requirement as well. All right, so we have some examples of some of the drugs where the testing is recommended, or sorry, the testing is required even before starting the treatment. So in here, we got 4 different drugs. So abacavir, carbamazepine. So then you have to really specify or do this testing before giving the drug to the patient to make sure that you don't put the patients in the extra risk of adverse effects. So we'll come up to the example of abacavir in the next slides. So this particular drug is a bit of interesting drug. So here in the first category, to avoid treatment in patients with certain genotypes, all right? So here we are looking at, we know like these drugs, a bunch of drugs have some certain adverse drug reactions in special genetic people. So people who are positive for example, abacavir or carbamazepine, HLA-B positive genotype patients, they show some severe hypersensitive reactions that could be even life-threatening. Same is with Raspberry case as well. Whereas when it comes to this drug, which is used for ivacaftor, which is used for cystic fibrosis, this drug is specifically useful in certain genotype patients only. So if you give it to some other genotype patients, or the patients which are not positive for CFTR, this drug may not be even beneficial to them. So there is no point of giving that particular drug. So that's where genetic-based treatment applies to these 4 drugs as of now. Is a recommended testing for these 4 drugs. So abacavir also is important for us where we're talking about COVID now. So there are some implications of usefulness of abacavir being an antiviral drug in COVID treatment as well. So that makes sense. Right. So let's synthesize the genotype guided use of abacavir a little bit more so that it will also shed some light on how the pharmacogenomics can be translated into practice and how this all, like what we know about pharmacogenomics now, how that kind of build up over the time. So I'll just point out to this meta-analysis of 13 different clinical trials. So this was published back in 2012, I think, which is almost 8 years now. So it's long. If you see, in a way, it's not a very new publication, but the idea already built up. So if you look at even the history of pharmacogenomics over the past couple of decades, it has matured much more. So coming to this study, you have 13 different studies. So, and they divided the studies in the different ethnic groups as well. So for example, Caucasians, white people, African American, Black, and I'll just ignore this particular study. Then Asians. So Asians, I think back of mind, I think these are Korean patients. So this is a forest plot, which will give you a summary of all the clinical trials in that particular area. So if you look at it, so just for white patients, which included, I think, 6 different clinical trials. So the average of that looks like the odds ratio is about 25%. 25 is the odds ratio, which is pretty high. So let me just tell you what exactly it is. What's the odds ratio they're talking about? So they're talking about the odds ratio of patients having hypersensitivity reaction with abacavir when they are known to be positive for this HLA-B5701 gene. So that means the patients who had this gene and what is the chances of them to have the hypersensitivity reaction with abacavir. So when we're talking about 24.331, that means the chances of having hypersensitivity reaction are 25 times fold more, which is pretty high. So it means a patient who is negative to this particular gene, the chances is 1, for example, or 0. Then if the patient has it, if positive to that particular gene, this is like 24 times more. Similarly, the same numbers you will see even for African American patients, pretty much similar. So more interesting is when it comes down to Asian. Now the risk goes almost 40 times more. So now we already know even like the prevalence of HLA-B, like number of patients who has this gene positive is more in Asians as well. It's more in Asians, plus it's also there is some sort of ethnic relation between the gene and the hypersensitivity reaction. So this is 40 times more, like the odds ratio is 40 times more for the Asian origin patients to have adverse reactions. So probably now it makes sense why the regulatory authorities has made this testing as a compulsion. And remember, it might just, whatever we're saying, 25%, maybe just about 6 to 7% of the patients, some of the ethnic groups, but their chances of having the life-threatening hypersensitive reactions is much, much higher than the normal people. So that's why it becomes significant. So based on these studies, now we have certain guidelines what to do when you're going to start the treatment with abacavir. So patient considered for abacavir. So first thing you have to do is perform the test. That's a genetic test. So see whether that particular patient is positive for this gene or no. If it is positive, stop. You don't give abacavir, or you use some other alternative medication. And if it is negative, still we don't stop there, because even some patients which are negative, they might still develop hypersensitive reaction, but the chances are less though. but they might still develop the adverse drug reaction. So you can give the patient the drug, but monitor the patient for hypersensitive reaction. So the patient doesn't have it, good. But if the patient have any adverse drug reaction to that particular abicavir, then you have to stop the drug, switch to something else. So this is some of the continuing recommendation in terms of abicavir to HLA-B genotype and phenotype relation. So non-carriers, the patients who are negative to the HLA-B gene, they are at less risk of having any adverse drug reactions because of abicavir. And the patients who are the carriers of this, definitely they are a significantly increased risk of hypersensitive reaction. And in that case, we will not give that the medication to the patient, or you can just use some other alternative treatment. Well, so that was about where there was the regulatory requirement to do the testing. There are certain drugs, again, I just came up in terms of FDA, I came up handful of drugs where the testing is recommended. So you have azathioprine and some of the drugs with that particular gene. You have to test whether the patients are positive or not, but still it is not a requirement, but it is recommended before starting the treatment. Then in terms of actionable pharmacogenomic testing, you have a bunch of other drugs as well. I listed some of them. And then there are some other recommendations where it's like not clinically significant. So the list is keep on growing. So again, you can also see like the liver enzymes are quite a few different enzymes here listed for different drugs as well. Well, if anyone wants the slides for this, they can have it for their reference. You can ask the organizers to get it from me. I'll be very happy to pass it on. Now, let's look at the genes implicated to affect the PD or PK of drugs used in COVID-19. So just a disclaimer here. When we're saying pharmacogenomics, we're not talking about viral genome. We're not talking about the different strains of viruses. We're not that probably get into the pathophysiology of the disease, but we're not talking about that aspect. So when we're talking about pharmacogenomics, I'm basically focusing on the genes of humans and their variability, how it relates to the variability of the drugs. So let's look at some of the drugs which I came up in my research, which in terms of genes and drug pairs that will affect the PK of the drugs. Well, we know like CYP3A4, which is a liver enzyme, it is involved in many different drugs, metabolism of many different drugs. So it accounts for almost 50 to 60% of different drugs. And we also now know like there are a lot of polymorphism in that particular gene as well, with variable effects, variable potency of the gene in different human subjects. So far we came up to some of the substrates. We know like these three drugs, darunavir, lopinavir, and ritonavir, these are the drugs which are also the substrates of CYP3A4. So we should have that in mind, like there is a polymorphism in CYP3A4, and the efficacy of these drugs might differ in different groups of patients. So I got I'll just get into the, just want to show up this particular table where you have different different polymorphisms of CYP3A4. So CYP3A4 star 1 is considered to be wild type, not exactly wild type, but like if you say the activity of this gene is 1, and rest other genes we just compare as to this particular gene. So most of the other variants of genes, they have decreased enzyme activity. Some might have same activity as well. For example, CYP3A4 star 10 is like considered to be the same activity of star 1. So, so what I want to focus here is there are so many different variants, polymorphisms of different genes of this particular gene with different enzyme activity as well. So when you talk about the drugs, which are the substrates of CYP3A4, we need to keep in mind that this can account for the variability as well. Other drug I want to focus is losartan. So people might say, why we're talking about that? Because there was some studies, still it's not a very conclusive study, which is still into the level of level 3 when you talk about the level of evidence, clinical evidence. Here it stands at level 3, which is like not very strong, but some of the clinical trials are underway. So once these are completed, maybe we will have more confirmed results on this particular drug. So losartan is a ACE inhibitor, and somehow it's like implicated to have some better outcomes for COVID patients, especially the pulmonary effects of the disease are like well controlled when the patients are given losartan. So as I said, this still has to be confirmed, but there are some implications for that. But for our topic, we know so losartan is a CYP2C9 substrate. So when we talk about CYP2C9, then there is also a genetic variability in the activity of CYP2C9 in different patients. So for example, you have the different allele of CYP2C9. So star 1, which is considered to be the wild type, so normal activity. But once the patients have the different copies of genes, say for example, star 1, star 3, so that means they are heterozygous for two different copies of a particular gene. Star 1 is the wild type, star 2 is the variant type. So these people will have compared to the wild type, will have less activity for the metabolism capacity for that particular gene. And similarly, as you go down, and these guys, for example, star 5 and star 6, they are both like there is not even a single copy of wild type. Both of them are variant types, so their activity will be even much less compared to other enzymes or compared to even star 1, star 3, star 1, star 5. So meaning to say, in CYP2C9, again, you have a lot of genotype variability. So ranging from like so far we know perhaps 1, 2, 3, 4, 5, 6. So sometimes even it will add up if you come to know some more later, or if you discover some other genes, we will come to know the more variability as well. So anyway, so this is about losartan. So how that can affect the activity of losartan when the patients have different genotypes of CYP2C9. Then so when it comes to the side effects, so now there's a different, talking about some different aspect on it. So we covered the PK aspect. So this is like we can say PD plus toxicity aspect as well. So here we're talking about the drugs which are potentially known to cause QT prolongation and ECG. And again, here we have, say, the drugs with different genotypes, like with the substrates of different CYP enzymes and the different genes they are affected by. So for example, CYP2D6 poor metabolizers. So say we have all these drugs. So you can see when it comes to poor metabolizers, so that the drug concentrations, the drug concentrations will increase and they might cause some adverse drug effects. So in that case, the usual recommendation is to limit the dose. You reduce the dose. In some aspects, you limit the dose. In some aspects, you reduce the dose. Same with other drug enzyme as well, that's CYP2C9 poor metabolizers or CYP2B6 poor metabolizers. So the similar line of action can be taken. Well, there's a lot more to say. There's a lot more to talk about when it comes to pharmacogenomics and different classes of drugs. So I tried to limit some of the drugs which are potentially be useful in COVID in terms of the PD actions and also in terms of the potential toxicity as well. So, but in the interest of time, I'll stop it now. If someone is more interested, I'm free to be contacted. You can get my contacts from the organizers. Like we can discuss about these things more if time permits later. Thank you very much. And but in that thing again, just going to the conclusion of the talk. So knowing pharmacogenomic can help in taking informed decision, especially when you target a patient, and then pharmacogenomics becomes important mainly for drugs with narrow safety margin, and drugs currently in use for COVID may not be the direct subject of pharmacogenomics due to lack of data, and drugs with side effect, like the drugs used in COVID are usually with a wider safety margin. Perhaps that's not an important aspect to consider as of now, but knowing the information is always the strength. So thank you very much. And I'll just pass it on to the organizers to proceed for the other proceedings. Thank you.
Thank you, Dr. Mohi. Thank you for guiding us about pharmacogenomics, basics of genes, to enlighten us about the important aspects which are going on right now in COVID-19 and about brief about your research. Thank you very much for that. Now we will move ahead with our talk. Our next speaker is Dr. Miraj Alam Khan. Dr. Meraj Khan receives his master's degree from Jamia Hamdard University, New Delhi, and his PhD is completed from AIMS Delhi. Later, he moved to Toronto, Canada, and joined Innate Immunity and Lung Laboratory at SickKids University of Toronto. Now, he has largely focusing on the understanding of the molecular mechanism of NEDs, which is nothing but neutrophil extracellular tract. So maybe this term might be new for many students. Okay, so you will learn more things about that and their role during pathological conditions and drug screening. So recently he extended his research related to COVID and lung pathophysiology, and as I earlier told you guys that his theme is topic role of neutrophil extracellular tracts during COVID-19 lung pathogenesis. So I'm connected you all with Dr. Miraj.
Thank you, Rajesh, for your kind introduction. I really
Thank you.
appreciate this opportunity to share my research and see, and in fact, sharing research will establish collaborations, and I'll get the feedback and suggestions. So let me know. And one more thing, I will be more focusing in the pathophysiology of the lung disease, and I will start from my research, what we did, what we found, and where we are moving forward. So I will touch towards COVID lung pathology, pathophysiology, drug screening, and to some aspects, personalized medicine. And recently, I started data science, machine learning approach in medicine. So I will share, I'll go briefly to that. Let me know when you can see my PowerPoint. Can you guys see
Yes, we
this?
can see,
Yeah.
sir. Yeah.
Okay. So as moderator mentioned, the topic today I'll be discussing the role of neutrophil extracellular trap during COVID lung pathogenesis. So we all know that neutrophils is the first line of immune defense. What does it mean? It means that that whenever there is infection, inflammation, or injury, neutrophils are the first innate immune cell that reaches to the site of action. And how what it does over there? So it simply phagocytose to kill, to eat up the bacteria or the pathogens, or degranulate. In the degranulation, what it happens, they kind of secrete some antimicrobial peptides to kill the pathogen outside. And recently, actually it's not recently, it was in 2004, the other phenomenon of neutrophils was discovered, and that was called as neutrophil extracellular trap. And what happened in that cases, neutrophils recognizes, senses the pathogens, bacteria, fungus, or even in the virus. And if they are not capable of eating the bacteria, for example, there is a multiplication of 10, means there are 10 bacteria and one neutrophil. So neutrophils cannot eat up all the bacteria. So in that situation, neutrophil senses the presence of many pathogens, and they decided not to go phagocytosis, rather to make nets. In that situation, neutrophils die itself. It's called as suicidal death, and releases their DNA inside out and form a net-like structure to cap. And the DNA already have antimicrobial peptides and pathogens to kill the bacteria. So that's what neutrophil extracellular exists. In fact, this phenomenon was discovered in 1991, but was not recognized. We recognized this phenomenon and named as NETs in 2004. So I can share one of my experimental video on top panel. What I did here? So I took the neutrophils from human healthy neutrophils, and the bacteria Pseudomonas aeruginosa, and stain the neutrophils with two specific dye: the regular Hooke's dye that goes into the nucleus and stain the DNA and gives blue color. The other one is green dye, that's called a cyber green, that also stain DNA, but they are not permeable to the neutrophils. So what does it mean? It means if we are getting the green signals, it means neutrophils membrane is compromised, or neutrophils release their DNA. And now you can really appreciate how mitosis happen. So initially, the most of the neutrophils are blue, and eventually, when the neutrophils perceive the signal or the presence of bacteria, they form nets, and you can see the most of the bacteria, the neutrophils, they form a nets, and that shows in green color. And the bottom panel shows electron microscopy images of the very typical nets, and the bacteria are entrapped here. So the coming to the more towards the clinic and in pathophysiology. So as a definition, nets are good in terms of innate immune function. They are very good in capturing and killing the bacteria. So, but as the knowledge came in, people done more and more research. So they found that no, nets are not always good. They are bad too. So if you can see, the first one is the wound. So nets are helpful in wound healing. They minimizes the wound area, they minimizes the further infection, and they are good in wound healing. But in case of cystic fibrosis, as Dr. Iqbal was discussing about the drugs, I work after, and the many other aspects, I'll touch those things in my subsequent slide. So this is this diagram is from our one of our review article. Here the nets are bad. Why? Because in cystic fibrosis lungs, there's lots of neutrophil influx happen, and those neutrophils make lots of nets, and that nets decreases the mucus in the lung, and that causes the problem in breathing. Initially, people thought this clogging is because of just dying the cells or neutrophils, but now this is confirmed that this clogging is not because of the extracellular DNA, rather because of the nets. And now clinician, they started treating DNAs so that they can clear out the lungs. Recently, as we all know, that COVID lung pathogenesis and pathophysiology, there's lots of cytokine storms happen. that leads the influx of neutrophils, other innate immune cells, and they die and they form a uh, cell dead debris, and that leads in further the leakage, problem in breathing, and lots of alveolar damage. There are reports, there are many, there are I think only two research articles that shows the presence of nets in COVID lung. So that is another direct relation with the nets, the pathogenesis, pathophysiology towards COVID. I will be touching in my subsequent slides. So what the purpose is that if we understand the mechanism of netosis, why in some cases nets. So I want to confirm, sorry, I want to just clear out one more terminology I'll be using. The whole process of the formation of nets as is termed as netosis, like we say apoptosis, necrosis in other mechanism. So the whole, if you understand the whole process of netosis, and that will be really helpful in terms of defining where nets are good, where nets are bad. And if the nets are good, we can make more nets, or we can prime the environment where we our neutrophils comes more and they make more nets to control the disease. Or in other hand, we can control the problems of nets. Now the based on the existing or latest update, nets are involved in many disease exacerbation, starting from infertility to autoimmune diseases, lung diseases, kidney problems, even in cancer metastasis. There are many articles that shows the presence of nets that helps in metastasizing cancer cells from one organ to other. So we need, there is an urgent need to understand the mechanism so that we can apply the knowledge and we can screen out some drug to control the net associated diseases. So coming forward, I will be very brief to say what approach we are applying. So we are taking cells from, like we are differentiating cells from cell lines like HL60 cell lines to make neutrophils. And we also taking neutrophils from healthy or the patient donor, and then isolate the neutrophils and perform various experiment to first of all to confirm whether they are neutrophils, then whether those neutrophils are responding for the nets assays, and then we do many more experiments that I will cover up during some result discussion. And now we are in a stage of translating the mechanism. So far we understand a bit of mechanism of nets, how neutrophils respond, what are the signaling, and how neutrophils ultimately decondense their chromatin and releases inside out. But at this point of time, we are testing some drugs in mice model. We are taking blood from patients with cystic fibrosis. So we have so far tested this lung phenomenon in mice blood, in asthmatic horse blood, and some pig model where we do the lung transplant. And the next, as I mentioned, the next or I can say ongoing aspect is for to do something related to personalized medicine. In this case, what happened, because getting neutrophils from donor or healthy uh, or patient, especially from the kids, because I'm working in the kids hospital, is very difficult. And out of and getting neutrophils from 5 ml or 10 ml blood is tough. So we are kind of very constrained in terms of number getting neutrophils, and we cannot perform large scale uh, drug screening experiment. So in our department, there is they are making ips cells, stem cells, and those stem cells are taken from a specific patient, whether it's cystic fibrosis patients, male or female at different ages. And those, so from those ips cells I started differentiating the neutrophils. So if I differentiate the neutrophils and characterize in terms of their response, in terms of their behavior against our different agonists, then we can use those large scale neutrophils for the drug screening. We already have some drug screening data that we can apply for those things. So that the purpose is to to differentiate the neutrophils from a specific patient or a specific gender or age, to understand the behavior of nets and screen the drug. And the last the domain which I started learning recently during this COVID lockdown, because I'm mostly working from home. So the artificial intelligence, the concept of artificial intelligence is not new. In fact, this is known before decades and decades. But now, because of the availability of the technology, the very powerful computer modeling machines, this AI is now playing a very important role, even it's in business or even in medicine. So we all are aware about the IHME or the COVID updates and COVID projections. So they all are based on artificial intelligence, based on the historical data, based on the different variates, different aspects, and that will help machine to learn and give a projection. So in our case, what we are trying to do. So I have done lots of transcriptomics and genomic profiling by using microarray. So the problem with for the clinician or the basic science researcher is they are unable to handle large scale data, the millions rows and thousands columns. And in that case, what I started to download the data from many repository where they already have submitted, and doing meta analysis, training my different models and machines so that we can identify some traits, some markers, or some target therapeutic options. So, so because of the time constraint, I'm not going to discuss all my major findings, but I will touch some basic aspects so that will help us to understand the mechanism of nets and netosis. So first of all, I'll be showing how we make nets in our lab. So this is again a single cell live cell imaging where with the single neutrophil stained with the Hooks dye and the cybergrain dye, and this neutrophil is treated with PMA. PMA is a pharmacological agent that mimics the PKC pathway and activate the reactive oxygen production, and ultimately helps to make the nets. So if you can see very nicely, you can really appreciate how neutrophils behave initially, and eventually the neutrophils chromatin decondenses and it's kind of rupture up and it blasts. That's how the neutrophils make nets. And so what we do whenever we have any drug or any agonist like PMA, as I mentioned, A23 is a calcium ionophore that helps in the influx of intracellular calcium, and the control with no agonist, and we take here positive control the detergent that disrupt the whole neutrophils, and estimate the release of DNA through the time point. So that shows the netosis kinetics graph, and here we can say how any agonist or drug is helping in making this or not. And these are the Western blot that shows the marker of citrullinated histone 3. I will be discussing in the model what is citrullinated S3 and how this help in making nets. So coming to the next aspect, as I mentioned, I have done lots of transcriptomics study back home in India, in AIMS. So I want to use that transcriptomic in neutrophils. My previous expertise was transcriptomics study um, um, in placenta or the implantation biology, but here I used neutrophils. And what I found that when I performed the transcriptomic study of neutrophils treated with PMA and treated with A23187, this is the calcium ionophore, that calcium ionophores activates many more kinases and transcription factor, and that helps to release the DNA, or that helps to decondense the chromatin and make nets. So the point is that uh, neutrophils is terminally differentiated cells. What does it mean? It means neutrophils is not going to divide or differentiate, or neutrophils is not going to need any new protein synthesis to survive or to perform their function. So why there is a transcription happening in neutrophil? Because neutrophil is going to die within few hours. So we further tested the use of transcription inhibitor and translational inhibitor. So what I did here, these are the confocal microscopy images. The first column shows the control neutrophils are only not treated with any activator or any inhibitor. So inhibitor is here is actinomycin D, that is transcription inhibitor, and PMA and INO4, as I mentioned, these are the agonist. Agonist means they activate neutrophils to make nets. You can see very nice nets images. They are counter stained with the red color is citrullinated histone, while green is MPO myeloperoxidase. These are the biomarkers of neutrophils to identify or to estimate nets. And in calcium iono4, you can see even more nets, and there are more citrullation of histone is happening. If we use the actinomycin D stops the neutrophils, or I can say actinomycin D suppresses the net formation. So it means that the transcription is needed, but whether the translation is needed or not, for that we tested the translational inhibitory drug like puromycin, and there are many more drugs. And what we found that when we use the translational inhibitor, they are not inhibiting the netosis. So based on that paper, we concluded that transcription is needed, but not the translation for neutrophils to make nets. And that makes sense because this is a terminally differentiated cells. It don't need any new protein synthesis. So the next question was, because it's more fundamental to understand why and when neutrophils decide to make nets or not to make nets, or whether to go for phagocytosis or degranulation. And this is more towards biological infection model. So here we use the LPS. LPS is a component of bacterial cell membrane lipopolysaccharides, and we perform different experiments to identify the role of, or to find a target that suits neutrophils to phagocytosis. So these are the various experiments. The carry home message here is that when we use the LPS, neutrophils make nets. It means neutrophils recognizes the LPS as a pathogen and make nets. But if you suppress the JNK is a very important MAP kinase that helps in signaling the presence of LPS, and that leads to the activation of different transcription factor, and at the end that helps to decondense chromatin and to make nets. So here the first two panels shows the response of LPS to activate JNK, and the 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 bottom panel shows the response of LPS to make nets, and here in the right panel that shows the response of the live pathogens like E. coli and Pseudomonas aeruginosa that also makes nets. And in the bottom is this is a model that shows how LPS is signaled through the JNK. So LPS, there are TLR4 receptors that senses the presence of LPS, and that activates the Taibah pathways, and ultimately then the activation of JNK that leads the reactive oxygen species production, and finally chromatin decondensation and nets release. So this is a model which we published based on these five major findings and many more. So so far there are two pathways of nets, or I can say netosis: the NOx dependent pathway and NOx independent pathway. As name suggests, NOx dependent pathway involves the reactive oxygen species through NOx, and they activate different kinases like ERK, AKT, P38, and SRC, and they also activates the myeloperoxidase and neutrophil elastase, and ultimately a sets of transcription factors, as I mentioned in my transcriptomics study, that helps to decondense the chromatin. And finally there is a protein called as gas dermin D. Gas dermin D is activated, that helps in making the pores on the membrane, and then the chromatin releases as a nets. While in case of NOx independent pathway, here neutrophils don't utilizes NOx ROS, rather they uses mitochondrial ROS. And because of the calcium influx, calcium intracellular influx, mitochondria ROS is activated, and that calcium influx also activate PAD4. So here is an interesting point. So PAD4 is a peptidyl arginine deaminase 4 enzyme. They are present in neutrophil cytosol, but in the excess amount of calcium influx, they activate PAD4, and the activated PAD4 goes into the nucleus, and they citrullinate the histone protein. So how it happened? So in histone there are arginine amino acid, and so this PAD4 removes the amine group of arginine. So if you see the structure of arginine and citrulline, both are same except the amine group. So when this PAD4 removed the amine group, then the arginine became citrulline, and because of the disturbance of the charge, neutrophil decondensation happens much faster, and that's why in case of NOx independent pathway, neutrophils go faster, or neutrophils make faster nets compared to the NOx dependent. And what we further found that this NOx dependent pathway is more towards or more in the situation of the infection or the inflammation, while this NOx independent pathway is more towards in the sterile injury or inflammation, sterile, like like in case of rheumatoid arthritis. This is in case of many lung infection or uh, um, and or any other infections. So other than these are the internal mechanism, we also discovered the external factor. I'm not going to share the data because of the time limitations. So we also found, because the hypothesis behind the effect of pH and salt was that neutrophils migrate from one organ to other through the blood, and they faces different changes of the pH. Like the lung pH is different, the gut pH is different, and the blood pH is different. So we studied a series of pH starting from like 6.8 more towards base basic to up to 7.8 more towards alkaline. And what we found that the in alkaline pH helps neutrophils to make more nets compared to the acidic pH. There is a lots of mechanism and the story behind that. And I did also tested the role of salt, because especially in case of lung infection, we always take the salt and we gargle. So I thought, let's test the salt and neutrophils, and the tonicity or the osmotic pressure of salt is doing anything or not. So we found that the hypotonic salt, they are kind of um, promoting the neutrophils to make more nets, but we don't have much mechanism in that aspect. So as I mentioned, that we found that pH could be a correcting method. Like for example, higher pH makes more nets, less pH makes less nets. So in a situation where, in a disease situation or pathology where we need less nets, so in that case we can correct the pH towards acidic, and may control the netosis, excess netosis, and vice versa. So the next question was, okay, so far we have more understanding of mechanism, and how do we translate? So in that aspect, it was in 2017 I was reading some article and found that, oh, nets are also involved in cancer metastasis. So that was kind of intriguing news for me, and I discussed with my supervisor, and then we contacted cancer MD Anderson Cancer Research Institute, and they provided us around 124 chemotherapy drugs, which are like very commonly used. And then what I did, that I tested all 124 chemotherapy drugs against four agonists. As I mentioned previously, PMA are the pharmacological agonist that make nets. LPS is a bacterial component which also activates nets. And here the other one, A23, is a calcium ionophore, sorry, and uh, the ionomycin is a bacterial component. So what so this heat map shows the the profile of those drugs, the effect of those drugs in against netosis agonist. And what we found that out of 124 drugs, there is a class called as anthracycline that suppresses netosis in all conditions. So it means, so whether the neutrophils are making nets through NOx dependent pathway or independent pathway, anthracycline is suppressing the nets. So these were the screening experiment where I only used one concentration of the drug. So I took this anthracycline and studied further. Anthracyclines are epirubicins, daunorubicins, doxorubicins, and idarubicins, and these four anthracycline drugs I did. First of all, I tested in a dose dependent response, whether their effect is really genuine or not. And if you can see, there is a dose dependent response of the suppression of the nets in LPS. I have tested in all other agonists. This is the only representative data, and these are the confocal images that shows the suppression of nets by these anthracyclines. We have studied the mechanism why this anthracycline suppresses not the other chemotherapy drugs, but that has many more data, and we published this in recently, I mean in the last year, in a cancer research article. And so this is a kind of our step towards translating the finding and and discovering some drugs which may be helpful in treating a disease which are associated with nets. But we have to test this in vivo. So for the in vivo, we are in the process of testing these drugs in mice model. So we have a mice model of lung infection, and very interestingly, what we do, we take LPS or bacteria and instill through the nasal. So it's called as intranasal instillation. And when those bacteria goes into the lungs of mice, the mice shows the inflamed or the infectious lung after 24 hours. And then we treat these drugs, whether by treating these drugs they are helping in their infection or not. And we take their BAL, it's called as bronchoalveolar lavage, and test the presence of cells and presence of nets. So this is an in vivo study. We still in the process of completing this in vivo testing of the drugs. So coming to the COVID and the present situation, as I mentioned in my previous slide, that nets are pretty much involved in COVID lung pathophysiology, or we can say pathogenesis. So here we can see this is a image which I drew recently of this is image of the single alveoli, the left side and the right side. So the right side is towards the infectious or the damaged lung, while the left side is a normal, the structure.
I think Dr. Miraj got disconnected. Some problem occurred in streaming. Anyways, meanwhile he's joining, we will go move ahead with the program because it may take some time sometimes. So our next session, we'll wait for 1 minute. Please be with us. If he's joining, then we will continue. Anyways, it was around 5 p.m. Sorry, 5 a.m. in Canada, and he was joined from there. Some issues. He got some issues in the net. So our next speaker is Dr. Nitin Saigal. So we'll move ahead with our program. That he is a senior scientist at Hilleman Laboratories, New Delhi. Mr. Nitin, Dr. Nitin is actually PhD in pharmaceutics and started his career from Dr. Reddy's Laboratories and then worked in Wockhardt Research Center. And he's presently working as a senior scientist in Hilleman Laboratories. He has he has patents granted in India, US, Europe, Russia, South Africa, Japan, Singapore. He has more than 220 patents are there, which are already under different stages of examination, and has authored 12 publications in peer-reviewed journal, which is a big achievement for someone who is from industry. And his topic of discussion is microneedles and audacious and transformative substitute to conventional parenteral delivery. So I'm connecting Dr. Nitin with us.
Okay, so
Welcome,
I
sir.
hope I'm audible right now. Thank you so much. Thank you so much, Rajesh, for this introduction, and thanks for having me in this beautifully organized short virtual conference kind of a meeting. So I'm quite happy and appreciate your efforts to conduct this as well. And I'm also happy to congratulate Dr. Mohi and Dr. Shailendra, who actually gave a very good insight of their work. And so I'm going to share my screen right now, and I hope it is visible, Rajesh.
Not now, sir.
No, it's not. So my screen is visible.
No, we are not able
Okay.
to see your screen.
Second. Let me try again.
You have to just click on share screen and then pop-ups will come. You have to select the screen.
Yeah.
Yes, now it
So
is.
is it now visible?
Yes, sir. It
Okay.
is
Okay.
visible.
Thank you so much. So, so to start with, just let me give a brief introduction about our company. So, I'm working in MSD Welcome Trust Hilleman Labs, and I have a total years of around 7 years association with Hilleman Labs. And so we are a joint venture between MSD from the US and Welcome Trust from the UK, who are 50-50 joint ownerships of this company, and we are based out of Delhi. And specifically on the aspects of developing vaccines for the global economies, and specifically developing vaccines for developing countries. And so we have a very interesting model to first take a particular technology to a stage wherein we can establish a proof of concept, and we can generate some sufficient data, take it to the level of preclinical assessment and clinical trials phase one, followed by technology, transferring the technology to an appropriate company, interested partners. So this is how we work. I mean, broadly, this is how we work. And so giving you, I hope you understand that, being in pharmaceutical company or industry, so we are limited by confidentiality as well as intellectual property matters to, not to limit our research, limit our data sharing on these kind of virtual conferences. So I have picked up a very interesting topic, and I'm going to give you a very broad overview, a very high level overview of this particular topic, because this is one of the subjects which has currently has gained a lot of momentum in the last 5 years. And this is one of the subjects in which we are actively working since the last 3 years, and generated sufficient amount of data so as to on the preclinical as well as the clinical studies which we have conducted. So the subject is microneedles, and I know the subject of the today's meeting is pharmacogenomics and personalized medicine. So I will try to link this subject, this microneedles to pharmacogenomics, as well as to the current situation of ongoing COVID pandemic, as well as to the personalized medicines. So just to give you a broad overview, what are microneedles? So I always introduce it by calling a band-aid a kind of a patch having very minute micro projections which are projecting from a substrate patch, and this is specifically used for intradermal applications of vaccines, as well as small molecules, proteins, contraceptives, hormones, and all. And this particular technology was developed way back in 1971 by Alza Corporation, but that was just the concept, and people started actually working on this once they realized the actual benefits of this particular technology. So it's basically meant for intradermal application, that is the application in the epidermis or the dermis. So the premise or the basis is to penetrate the stratum corneum layer of the skin and to deliver the drug below the stratum corneum, that is in the epidermis or the dermis part. Specifically, it is very useful for vaccines delivery because of the fact that there are a lot of antigen presenting cells which are also called as dendritic cells. So which have the responsibility of presenting the antigens to the immune system in a, I mean, in a very basic language I'm talking about. Because there is a lot of there are a lot of antigen presenting cells on the surface of the skin below the stratum corneum in the epidermis and dermis. So that makes particularly this kind of a delivery very relevant for vaccines. And as we move on, there are a lot of solid, hollow, dissolvable microneedles, and they usually have an insertion depth of somewhere close to 1,000 microns. Uh, and the best part is that these are painless, these are prickless, these are phobialess, and there is no pressure in applying the needle, because we all know how much difficult it is to administer vaccines to infants and to kids. So, and why I have mentioned this as a transformative technology, because this has the potential to completely transform the way parenterals and vaccines are delivered. But since this is a new technology, this is a very this is a statement which is quite early, because a lot of work is going on, and we might definitely see a lot of commercial products entry into the market in the coming 5 to 10 years time. So, so giving a basic overview of what microneedles are. So basically, there is a band-aid kind of a patch which has to be applied onto the skin, and the patch has two surfaces. One has an application surface that's completely plain from the above side, and there is a micro protrusion surface which have multiple microneedles which are projecting from that patch. And the best part is that the microneedles sometimes can also be made up of drugs, or they can be made up of a solid material like a stainless steel or a titanium, over which the drug can be easily coated and applied. So it's just the deposition, it's just the mechanism of depositing the drug below the skin. And since these biomolecules are themselves not able to penetrate or permeabilize the stratum corneum, so this particular technology is very useful for depositing the high molecular weight biomolecules below the skin. And so these can be classified basis upon the material and compositions, their applications, design, and the manufacturing methods. So material of composition, as I told you, it can be polymeric needles, which are completely made up of polymers. There can be solid needles, which can be precision coated with the drug. And based upon their application, the applications can be for specifically delivering vaccines, for delivering small molecules, for delivering hormones, contraceptives, supplements, iron supplements, or they can be multiple applications. So, and the design, I mean, the figure which is presented in this slide is basically how we classify the microneedle based upon the design. So the design is such that there can be a solid removable microneedle. As you can see on the right side of the slide, there can be a solid removable microneedle in which the microneedles only cause some perforation in the skin, and once you apply it, you remove it. There are voids created below the skin or below the stratum corneum, and then you apply a patch or a band-aid kind of a patch for depositing the or penetrating or permeating the drug onto the skin where the voids are created. So this is one of the methods how we deliver drugs by which are unable to penetrate the stratum corneum by their own. And the second is the coated microneedles. As I told you, these can be made up of either stainless steel or titanium or any other compatible metal, which can be easily dip coated or precision coated or I mean there are multiple methods of coating of the skin, but this is a very precise method of coating the microneedle. And once you apply it, the needles, they cross the stratum corneum layer, and they deposit, or the drug gets dissolved below the skin in the epidermis or the dermis part. And since the drug or the vaccine is getting deposited just below the stratum corneum, where there are no nerve endings, that is the reason why you do not reach that area where there are multiple pain receptors. So this is definitely a painless kind of a delivery, because when we work on this delivery, I have applied our microneedle patches multiple times onto my skin, and believe me, these are absolutely painless, and very precisely they deliver the drugs below the skin. So that's the reason why I call them as a transformative and a bold technology, which can actually replace the conventional parenteral administrations. So talking about the second part, which is the dissolvable microneedle part, in this, the entire needle can be made up of a polymer in which the drug is encapsulated. So this is quite interesting because of the fact that these solid or coated microneedles can still be having some kind of a sharps or sharps waste, but these dissolving microneedles, since the microneedles are made up of polymer and the drug itself, so once you penetrate, once you apply it, the drug as well as the polymer gets dissolved below the skin, and there is nothing. You only are left with a patch, which can be simply thrown away. So the best part about the polymeric needles is that these needles can be these are strong enough to penetrate the skin, but they are weak enough to let the drug gets dissolved below the skin. So this is the beauty, how we optimize the polymeric needles, because they have to be kept strong so that they are stable, they are easy to apply, they penetrate the skin barrier which we are intending to penetrate, and at the same time it has to deliver the drug. So then hollow microneedles are something in which, I mean, these are not the ones which we are working on, and these are not the ones in which we specifically have any focus on, because these are the ones in which we apply, and then through pressure we apply the drug. So this is a concept which is not very well worked upon. So I'll skip this maybe. And then hydrogel forming microneedles are something which are, as we know, in tablets also we have immediate release, sustained release, modified release, or extended release. So in this particular type of a concept, we have hydrogel forming needles in which sustained release polymers are used for forming or deposition of the drug below the skin, and then these hydrogels, because they are slow releasing, so they slowly release the drug into the systemic circulation, and these are specifically used for contraceptives, because they require somewhere around a 5-day course to be done. So need not to take pills every day, and simply you deposited the drug and then it can be released. So this, and talking about the manufacturing methods, there are multiple manufacturing methods, because this can be it requires a lot of engineering expertise. I mean, it actually is a amalgamation of engineering, biotechnology, formulation science, composition science, so everything clubbed into one. So as far as I talk about the manufacturing methods, uh, the the needles are actually made up of electric discharge machining or micro machining processes, which are specifically linked to the engineering concepts. And so we decide upon how the which antigen which has to be incorporated, and potentially all the vaccines, and most of the low dose drugs can be definitely be included in this technology for our administration. And there are many methods available in the literature, like droplet air blowing, in which you droplet to form pull a micro droplet to form microneedles from both the sides. So there are many methods by which we are available, by virtue of which you can make microneedles, but this involves a lot of expertise, as well as precision based expertise, which is required for manufacturing those. And so, as I talked about DMNs, which is dissolvable microneedle, these are fabricated with the biodegradable polymers, and the drug gets actually encapsulated. You can see the red part is the drug or the antigen, which gets encapsulated into the polymer. And once the entire needle polymeric drug encapsulated polymeric needle, once it gets administered, then the dissolution takes place, which releases the encapsulated molecule into the skin, and you can simply pull out the patch, which has to be anyway discarded. So, so particularly these are used, for administration of vaccines. But as I told you, this is quite broad. I mean, this has a lot of flexibility, which I'll discuss in my subsequent slides. The potential value, uh, as I told, I'll be just going very broad, and I'll definitely welcome any post-presentation questions, as well as the contacts. If somebody wants to get more details, definitely you can. There are my details at the end of the slide, so you may note it down, and you may contact me anytime. But I am currently not presenting any specific data on which we have developed, which we have generated on microneedles, because of the limitations I have. But giving you a broad overview, what is the ease of administration? Because as I told you, these are painless, these are phobia-less. There is no pressure of delivery, and it's simply application of a band-aid, pressing it for a couple of minutes, and you are done. So, so, so depending on the how the regulatory acceptance is done, this has a lot of potential for home use of vaccines. And if there are some vaccine experts, or if there are some experts in the audience, they might realize that, for example, for rabies, if somebody is bitten by a dog, then the course of administration for rabies is very complex. You have to have multiple doctors visit, and you have to have multiple administrations of injection. As per the WHO's recent rabies administration schedule, you have to go at least 5 times and apply the injection twice a day. I mean, there is 2 2 0 2 2, which means you have to go at on the day of bite, then on the 7th day, then on the 14th, 21, and 28th day, and every day you have to get at least 2 injections done. So, but many times, in the rural areas, the people, because this is a life-threatening disease, so many times the people, they die from this disease because of the fact that they are unable to complete their immunization procedure or the schedule. So this particular technology has a lot of potential for home use, because of the fact that it requires minimal training. You don't have to have a registered medical practitioner, or a person who is expert in delivering the vaccines, and this can be used for home use. And this is also having excellent user compliance. As I said, minimal training is required, and there is a potential of an OTC product as well. As far as the thermostability is concerned, I hope you all will understand that for vaccines, particularly, we require the vaccines to be thermostable, because in a country like India, where you have to have maximum outreach of the vaccines to the remote areas, and in many of the areas there are no refrigerators, or the there is no procedure of storing the vaccines at appropriate place, and that's the reason why they degrade. So you need to have thermostability, and since these microneedles have the drug which is in the solid form, and there is a scope of inclusion of a lot of stabilizers as well as an excipient in the microneedles. So there is a potential of vaccines to be licensed as a CTC, which is controlled temperature condition use, which is usually more than 3 days up to 40 degrees. So this is also one of the advantages. There is a rapid onset of action. It avoids for the drugs. It avoids GIT first pass, and the for the vaccines it has enhanced immunogenicity because of the presence of antigen presenting cells below the skin. And if we talk about the programmatic advantages, I hope you all will understand that it's not about only the cost of developing a vaccine. It's also about the cost of logistical arrangements which the end user, or the end take care person has to provide. So in case if you are replacing a parenteral vaccine by a microneedle vaccine, Then first of all, there's elimination of cold chain, logistics become simplified. There is a improved presentation, reduced footprint, and then you don't have to have many sharps for disposal. So overall, it's going to reduce the burden on the healthcare systems, and obviously, since these costs are associated with the programmatic usage of all the vaccines, so it's definitely going to cause an overall reduction in the public health expenditure. And of course, if we talk about outreach, there is a very popular example of measles and rubella vaccine, which is actually not able to reach the wider population because of the administration issues, and the reach and the outreach has been stagnated to almost around 85%. And so people are taking this MR, measles rubella candidate, as a very good candidate for being integrated with the microneedle patches. And as I told, I mean, ease of administration is also linked to campaigns, because why the I hope you all understand with that how why the oral polio vaccine has been a success, because it's an oral vaccine. First of all, I mean, there are a lot of government efforts in that, but since this is an oral vaccine, so the house to house campaigns for these vaccines was quite easy. Otherwise, if it would have been a parenteral vaccine, then again, I mean, the eradication would have been a lot more difficult than it has been right now. So, so these kind of microneedle delivery will also enable house-to-house administration of vaccine, because of reduced logistics, and and obviously because there are no cold chain requirements. And one of the very important thing of this particular delivery technology is the dose pairing. So dose pairing means that whatever immune response you get from a complete intramuscular administration, for intradermal through microneedles, we have seen the similar immune response with 1/10 of the dose or 1/5 of the dose. That depends upon individual antigens, but the same immune response is coming because it's intradermal delivery, because of the antigen presenting cells. The same immune response comes through 1/5 or 1/10 of the dose. So it actually translates to the cost of the vaccines. And as far as applicability is concerned, these are very there is a very broad applicability to almost all the parental vaccines, and also it might facilitate the novel vaccine combinations as well. So, just to give a broad overview of how these microarrays are fabricated, first of all, it requires the intervention of an engineer to make master molds and to make micro molds. I mean, you can just imagine how we make ice cubes in the fridge. So it's just that we need to have a micro needle mold, so as to be when we pour the polymeric as well as the drug solution into those molds, it gets dried, it gets solidified, and then the polymer solutions are casted into the micromolds, and through the negative pressure as well as through centrifugation, we remove the air pockets or the voids which are formed during evaporation. These are allowed to dry. I mean, there is a backing membrane casting, because you also need to have a supportive backing membrane. And then these prepared microneedles are peeled off from the micromolds, and basically we are working on a robotic peel-off mechanism in our company for removal of these micromolds. So overview, so broadly, this is the step, but there are multiple optimization and fabrication considerations to be done, to be taken care of with respect to the length of the needle. I mean, it will depend upon how much is the active payload potential of the needle, so that how much active can be loaded. So, and also it depends upon the geometry of the or the flexibility and the resistance of the skin, that how much length of microneedle we need to keep for a specific antigen. And one thing I want to mention that I mean, it's not a one size fits all approach. I mean, every antigen, every drug will require a different set of optimization parameters to be done. So, so we have to work separately. I mean, this can be called as a platform technology, but actually, each drug requires a separate, separate target product profiles (TPPs) to so as to fit that particular criteria. So we have a number of microneedles per pack. We usually have around 14 by 14 or 20 by 20 array when you're accommodating around 196 to 200 needles per patch. And these are so small, these are not actually visible through naked eye. So it's usually, we usually use it through either look at them, look at the geometry through a microscope or through a magnifying glass. And then size of the patch, it depends upon the active requirement. And spacing between individual microneedles is also a very important parameter which has to be considered. And when we talk about spacing, when we talk about base, there is a very important term called as the aspect ratio, which is the ratio between the base and the height of the microneedle. And I mean, the multiple factors, and one of the important factors is concentration of active. I mean, what we usually do is we concentrate the active ingredient or the vaccine or the antigen or the drug to the tip of the needle, so that there is 100% administration and nothing remains, because usually because skin is also some kind of a resistance. So the active is I mean, the microneedle do not penetrate completely in the skin. So mostly 50% or 60% of the needle, they penetrate the skin, and the remaining they remain with the backing layer. So we have to concentrate the active on the tip portion. Then the drying time is an important parameter, that what type of excipients you are using, which depends upon how much quickly you want to administer a drug, or how much uh, strength you want for a particular needle. And then the concentration of excipients. I mean, these are just the broad parameter which we look at uh, for uh, for fabricating and optimizing microneedle. So primary and secondary packaging is also important, and then obviously you when you apply the needle, you need to have a specific either an integrated applicator, or you need to have a separate applicator which is broadly used for applying multiple patches. So what are the applicator requirement? What is going to be the design of the applicator is also quite important. Further, the application time. I mean, what we have developed in our labs is we have an application time of somewhere around 180 to 240 seconds, in which the patch has to be applied for complete delivery. But if you look at the literature, people have application times ranging from 1 minute to 30 minutes as well. So this is also an important parameter to be optimized, and this gives an edge over other competitors, because the minimal application time you have, the better it is. And what are the critical quality attributes which are identified? And this is courtesy conference which I attended in January, which was organized by PATH, which they have an MEP Center of Excellence and a regulatory working group. So, so these physical, I mean, these are all the parameters which we also evaluate, like the needle geometry, puncture performance, physical stability, how much is the wear time, how much is the mechanical strength, and how much is the force required to break a particular needle. So everything is, I mean, some are linked to engineering, some are linked to formulation. So this is a collaborative work which we usually do in our labs. And moving ahead. I mean, in the interest of time, I'll simply move to the applicability. As I initially mentioned, that these are applicable for vaccines, specifically for vaccines, because of the fact that there is a potential of thermostability, there is a potential of dose sparing, and then vaccines are mostly administered to kids. So that is the specific population which is the most which is having the maximum phobia of needles. So it applies well. It applies well to vaccines, and then we can also have it for peptides, proteins, and hydrophilic low dose small molecules. I mean, these are just some of the categories which I have mentioned. I mean, these are not limiting to the application, but I've just mentioned some of the categories in which this is mostly suitable. And why I mentioned low dose small molecules is because of the fact that this technology is limited by um, by the payload, because these microneedles are so small that you cannot really think of administering or integrating an antibiotic through a microneedle, because antibiotics are usually high dose. So the limitation which we have derived is somewhere around 1 milligram per patch. But even if it is more than that, you can apply 2 patches or 3 patches at a time. So that's quite flexible. So immunomodulators, one of the good categories, which like I think Pidotimod is a good candidate. Imiquimod is a good one because they modify the immune system, and this is specifically linked to the current situation. And talking about COVID, coming to COVID vaccines, we are actually talking to some companies who are interested to to integrate their antigen with our technology. But if you ask about my personal opinion, I think we usually integrate the existing vaccines or registered vaccines with a new technology. A new antigen with a new technology becomes a little complex in terms of regulatory approvals and many other things. So it's definitely a good candidate, but yes, I mean, I think initially we should go ahead with the conventional delivery, and then once you reach to a problem where there is limited outreach, then this kind of a delivery system can definitely take over. So, but microneedles for COVID is a really hot topic these days because almost the entire world needs microneedle needs COVID vaccine. But definitely, once we have an antigen, once we have a registered vaccine in hand, the delivery is going to be a major challenge because in a country, even if you talk about a country like India, I mean, it's going to take years for the entire population to get vaccinated. So this kind of an intervention, this kind of a delivery system can definitely play its a vital role for a definite outreach. And specifically on hormones and contraceptives, as I said, a lot of hormones like a parathyroid, thyroid hormone, which are low dose, and contraceptives I have been used for this kind of a delivery system. And then multiple supplements like iron, folic acid, zinc supplements are also have been used for this kind of delivery. But I mean, these are just some of the these are just some of the examples which I have come across through literature. But if you talk specifically about our work, we are only focused on vaccines. And so coming to the, as we call it, as a can of worms, because it's definitely not a very straightforward kind of a technology. And people have been working for years, but there have been a lot of questions on sterility, on manufacturability, because eventually, if you are talking about replacing conventional parenteral administration, then sterility has a vital role to play. So whether the microneedle patches have to be manufactured in a completely sterile environment, or it has to have low bioburden, or it has to completely its hostility is not important. So these are the questions which we normally discuss in our meetings with WHO and PATH, and then we come up with the we share different ideas. And so this is something which is uh, matter of detailed discussion. And then coming to manufacturability, so some of the companies they have been able to take this to the level of a pilot scale facility, but since there are different methods to manufacture, so it is also very the large scale facilities are also quite limited. So since this particular technology has reached to a level of phase 2 or phase 1 studies, where we need a GMP manufacturing lab, so that we are actually quite limited by this. But having said that, a lot of companies, including our companies, working on scalability and manufacturability. And once we but that happens once we take over from the proof of concept stage, which we have successfully done. And so we are thinking about large scale manufacturing of this. And talking about clinical and regulatory guidelines, again, we usually have a lot of meetings with the policymakers and the regulatory experts to have concrete guidelines on clinical and regulatory path to taken forward, and then which is linked to how the product registration path is going to be. And lastly, talking about the transformation and investment, as the audience will understand that this is this technology has a potential to completely transform the way parentals are administered. And it becomes a little tricky for people like us even if we have a POC in our hands, it becomes a little difficult for us to convince the large scale manufacturers like Pfizer and GSKs to adopt this technology right away, because it requires a lot of investment, and they already have invested in their parental facility. So it needs a pull or a push, I would say, to for them to move towards this method. But considering the advantages and considering the trigger which we get from the I mean, multiple policymakers and people to whom we speak to, this is definitely going to happen. I mean, I'm quite confident that this kind of delivery is definitely going to either parallelly join hand with the current delivery mechanisms, or can they have the potential to replace it. And while we think of scalable manufacture, it is very important to understand that manufacturability or scaling the technology, it actually is a process which we have to think right from the day one, right from the day where we are when we are developing proof of concept studies and evaluating in animals. So manufacturing has to be taken care of, thought of. So the commercial product have to be recognized early with the mitigation of the development risks. And since this is a novel, bold, transformative, disruptive technology, it will require novel solutions to execute. So having said that, I would end my presentation here, and this was some references which I had referred while making this presentation. And with this idea, I am going to thank the organizers and the team which has given me this opportunity to present. Uh, I mean, not my work, but the concept in which we are working on. And I'll be very more than happy to have any questions.
Thank you very much, Nitin sir, for guiding us about your the concept of your research. And I think everyone got it very perfectly that how actually industry performs. This is very important for the students to understand that how actually the concept which they are reading or they are listening from this type of conferences, how actually the work is going on at the industry level. We will take just two questions because time is almost very much is taken. So my first question is for Dr. Meraj sir. How the novel role of mitochondria in neutrophils was defined by your work? How you did that?
Yeah, actually, that's a very good questions, and in fact, we made a very path-breaking research in this field. So, as I mentioned, actually, I didn't get chance to mention. So, uh, it is known that neutrophils uses glycolysis to for their energy production, not the mitochondria, which is a more efficient and more energy way. And there are reasons why not neutrophil is using. There are some mitochondrial factor are not there. So the next question is that then why there are lots of mitochondrial neutrophils? Since neutrophils is not using mitochondria for the energy production, they are using glycolysis. So when we identified the NOx independent pathway, then we found that neutrophils uses mitochondrial ROS for net for netosis. So what does it mean? It means that neutrophils is innate immune cells, and for innate immune function, nets neutrophil uses mitochondrial ROS. So we define a novel role of mitochondria, not the energy production, rather innate immune defense in neutrophils. That was a very interesting finding, and we published this paper in this research in PNS in 2015.
Thank you very much, sir. The next question is to Dr. Nitin sir that how microneedles can be cost effective if they can for COVID-19. This came from the audience. They wants to know that how and why.
Oh, sorry. sorry, I was muted. So we actually don't know the cost of COVID-19 vaccines as of now, how much it will cost, and but but we have done some cost analysis in our company, which is the cost of goods analysis, and what we have evaluated through various business experts as well as discussing internally that the I mean it. I mean definitely. I mean if the cost is not an advantage, then it will be difficult for us to convince the policymakers and the manufacturing people in the industry. So we did a COGS analysis, and we saw that it's actually either at par with the conventional administration. I mean, the cost of a vial, a syringe, and an injection is actually comparable to the cost of the, the logistics or the raw materials which we are using for making a microneedle patch. So this is about the cost of the raw materials which are used for making patches. But if we talk about the cost of antigens, then this is quite specific to the antigen manufacturers. I mean, obviously, all the antigens do not have the same costings, so it depends. It depends, but definitely it has the potential to be cost effective as well.
Thank you, sir, for answering that. The next question is for Dr. Mohi. That what are the challenges for implementing PGXs in practice?
All right. Thank you. The challenges, the main thing I would say is the awareness among the clinicians and especially the clinical pharmacokinetics, kinetic people as well. Yes, cost is also a bit of a factor as of now. So mainly the translation of research into practice, that's a main barrier as of now. And that, of course, as I said, the awareness is these are the challenges. I would say two major challenges.
Okay. There is also a question from audience for Dr. Meraj sir, that how you exactly isolated neutrophils. It was actually quite a topic of discussion amongst our audience, that how exactly neutrophils were isolated from human blood.
Yeah, actually, that's because I know this is a tricky and kind of tedious process, but because we do as a routine basis, so I can say it's not that tough. There are, in fact, kits available through in the market to isolate the neutrophils, but sometimes the neutrophils isolated through kits, they are kind of activated because neutrophils are very fragile. So we use very simple density gradient method, and we use per call so to make the density, and based on the density, neutrophils settle down at a particular band, and then we'll take that band and then we'll purify. So it's a kind of I can say it takes around 2 hours, 2 hours to 3 hours to isolate neutrophils, but it's not a, like that tough process. There are commercial available kits, and we also do manual.
Okay. And the next question is for Dr. Nitin sir, that what is intellectual property landscape of microneedle products? Just the question of IPR.
Well, so, yes, I understand. So as I told, I mean, the first microneedle manufacturing was done by Elsa Corporation way back in 1971. So there is a freedom to operate on the concept as such. But if you talk about specifics, there are different buckets in which the patents get filed. I mean, the one of the buckets is the composition in which we can talk about various polymers and excipients and antigens. And then second is the fabrication method. There are a lot of IPs on the fabrication methods available. And the third and the very critical one is the design of the needles. So some they have some of the one of the critical factors is the design of the needle, because you need effective penetration. You need effective penetration, and you need effective dissolution of the antigen below the skin. So if you specifically talk about the design, some have pyramidal shaped needles, some have cylindrical needles, some have conical, and some have a hybrid model. So So basically, to answer your question in short, there are around four buckets in which we can classify the IP landscape. One is the composition, the other is the design, the third is the fabrication methodologies, and the fourth are the specific antigens. Thanks.
The last question is for Dr. Mohi, that will there be any instances that pharmacogenomics test doesn't fit in the presumed correlation between gene drug effects?
Yes, that could be possible. Like, for example, we say a patient falls into one genotype category and his effects are so and so, but that might not be the case in reality. So we call the concept as phenoconversion. So where the genotype doesn't stands out as much, there could be some other factors. For example, maybe there's some other drug given along with this drug of interest, or could be some environmental factors, could be some other factors. So yes, that could be a possibility in clinical scenario.
So thank you very much for the panel discussion. We are not going ahead. There is so many things that we can actually discuss, but there is always a time limit and we have to go with that. With this, we are concluding our session, and at the end, I want to thank our chief guest, Dr. Sailendra Saraf sir, first of all, who gave us a time for this event. Then I want to thank our speakers, Dr. Nitin Saigal, Dr. Mohi Iqbal, Dr. Meraj Khan sir, and at the end, I also want to thank IPF International Pharmaceutical Forum for connecting with this event and jointly organize this event with PharmaTutor. Thank you very much to the audience, and definitely you will get certificate for the this event in your email within 1 or 2 days. Okay, so do not worry about that. Like, share, and subscribe with us, and always be with us. Thank you very much. Thank you.